Okay, so as I was saying that now we are on the end of the methylation topic, and here you can see there are two bases. One is the cytosine base, which itself at times gets automatically deaminated to produce the uracil. Now, uracil is not a normal base of DNA. And if we look at methyl cytosine, you can see that the methyl cytosine is spontaneously demethylated to give thymine. Now, if it is a cytosine base, spontaneous deamination occurs within the DNA, converting the cytosine to uracil. And if it is a methyl cytosine base, spontaneous deamination converts the methyl cytosine to thymine. Now, what is the problem? When cytosine is converted to uracil, we know that there is the repair mechanism where uracil DNA glycosylase recognizes the uracil and accordingly it efficiently removes the inappropriate base and initiate repair to restore C in place of U. So if that U is recognized, that U is restored by the original C. But it sometimes happens that when 5-methyl cytosine is automatically deaminated to give rise to thymine, thymine is a natural DNA base and seems to interfere with the efficient repair of lesion. Even though there is a thymine glycosylase, but this repair mechanism is not that efficient. And with the passage of time, the T tends to remain, the T persists, and the C to T transition persists and is passed on to the next generation. So it is evident that when there is a methyl cytosine deamination event is occurring, with time, ultimately a C to T transition is occurring, as a result of which the original base was CG, gets converted to TA. So this happens and this accumulates with time. Hence, methylated cytosine are considered as mutational hotspots because deamination is a normal chemical phenomena, and by this normal chemical phenomena, sometimes in the cases of methyl cytosines, ultimately thymine are induced. So this is one example where the methylation actually creates a drawback and introduces mutations within our genome. Now moving on to another example where the cytosine methylation actually serves as a system of defense. Now this basic mechanism where the methyl cytosine is being deaminated spontaneously to give rise to thymine, this property has been used as a defense mechanism. Look at this example. This is an invading DNA having a CG dinucleotide. Moment the invading DNA interferes and gets cells attached into this blank space, there automatically the C are methylated by the de novo methylases. And as you understand that in course of time, and this area, as I said previously, in today's morning class, we have seen that there are certain proteins which actually remain bound to the CG islands and protect the unmethylated CG islands. So this is protected. Now, when this the invading DNA is inserted in the genome, the de novo methylase enzyme methylates the Cs. As a result of this methylation, now these are methyl cytosines, which in course of time will get converted to T by the spontaneous mechanism of deamination. As a result of which, the T gets inserted in this DNA that has this foreign DNA. And as a result of which, the foreign genes will automatically in course of time be silenced, and mutations will be inserted in them in a permanent manner when the methyl cytosine gets converted to thymine. So this is how at times whenever a foreign DNA gets inserted, in most cases it is seen that the foreign DNA gets silenced. Now we next move on to the next slide. This is a case of differential genome imprinting. So let's look into this example. This is a beautiful example. See, this is the maternal chromosome and this is the paternal chromosome. Now, this is regulation of gene expression by DNA methylation. How does it do? So this chromosome has two genes, IGF2 and H19. This chromosome is present in a female body and the same chromosome is also present in the male body. Now let us look in the female body what happens. In the female body, there are two, as I said, there are two genes, IGF2 and H19. And there is a ICR element. This is the imprinting control region. And in this imprinting control region, we can see that CPCF is a molecule. It is a molecule. It's a regulatory molecule which can bind to this imprinting control region, as you can see here. Now, in case of maternal chromosome, this imprinting control region is not methylated. Since this portion of the DNA is not methylated, these proteins can easily bind with the ICR. The moment these proteins binds with the ICR, a problem occurs. What is the problem? I said there are two genes, IGF2 and H19. Now there is a downstream enhancer element which is needed for switching on of H19 gene or the IGF2 gene. So this is an enhancer element which simultaneously controls the H19 gene. It's a regulatory element for the H19 gene and also the regulatory element for the IGF2 gene. Now, in case of females, I said that in the imprinting control region, this this molecule has got itself bonded. As a result of which, the enhancer molecule, the regulatory elements, the enhancer at the enhancer region, the proteins bind, and they control the gene which is sitting adjacent to it. They cannot control, they cannot exert their control to the distant IGF2 because there is a sort of wall standing here. So in case of females, since the imprinting control region is not methylated, as a result of which H19 gene is expressed. Now let us look into the males. In case of males, it is the same chromosome. But interestingly, you see that the ICR region along with the H19 region is methylated. The moment this portion is methylated, as we know that none of the DNA binding proteins can bind to them. So what happens? Now this enhancer and the regulatory elements can exert its effect on the IGF2 and the IGF2 gene is switched on. So as you can see, it is the same chromosome. Depending on the pattern of methylation, it is switched on when there is no methylation, and IGF2 is switched, H19 is switched on when there is no methylation, and IGF2 is switched off, and when there is methylation, IGF2 is switched on and H19 is switched off. So this is a case of differential expression of two genes sitting on the same chromosome in a female setup and in a male setup. Now, next we are moving to another example where methylation, before going to that example, wait. Okay. Now let me show you some pictures of the molecules that helps in protection of the CGI islands. For example, these are METHYL-CPG binding proteins. There are certain proteins which can directly bind to the CGI islands and prevent methylation. That is one group of proteins. This is another group of proteins. These are, as you can see, these are METHYL-CPG binding proteins. That is, these proteins can bind to the methyl cytosines. So they recognize the methyl cytosins, and they are a group of proteins. And if you look into their domain structure, you can see that they all have a common METHYL-CPG binding domain. So these proteins also play a big role in repressing of gene expression. So we'll be coming to how these proteins play a big role through one of the examples, which I'll be talking on later on. And these are some detailed examples. These are all basically transcriptional repressors. As you can see, first of all, methylating a segment of DNA as or automatically makes it inaccessible to transcription factors. Further, if there are now proteins also attached to that methyl group, then you can understand that the DNA is totally inaccessible. So presence of these kinds of proteins further represses or silences a gene. So these are a group of proteins that play an important role in gene silencing. These are some techniques used for genome-wide DNA methylations. I'm not getting into the details of the technique. I have told you to go through the techniques. In fact, I had sent animation of this particular technique. This is a very famous technique, a bisulfite technique. So go through it if you have any problems related to the techniques, because the techniques are not exactly a part of your syllabus. But you need to know these techniques if you are to sit for the National Eligibility Test exam. So go through the techniques. I'm not getting into the details of the technique today. Now coming to DNA methylation and diseases. So this is one gene, as you can see, and this is the promoter element. And in the earlier class, I had said that about 60% of human genes have CG promoters. So they have CG islands. Now, this particular DNA has a trinucleotide repeat sequence. If you remember, we talked about the STRs when I did my genomic class, the importance of the trinucleotides. So this is basically a short tandem repeat. So it's a three sequence repeat, CGG, which has been repeated 6 to 59 times ahead of the gene, which codes codes for the FMR1 product. Okay, so the gene is FMR1. So you can see that at the promoter region, there is 6 to 59 repeats of the trinucleotide CGG. That is normal. The island is normally unmethylated, because I told you that in most cases, the CGI islands are protected from methylation. And I had given you in the morning an example that some DNA binding proteins, so having a CXXC motif, can bind to such unmethylated islands and protect them. So this is basically an unmethylated island and it's protected. Now, if there is a mutation by the mechanism of replication slippage, hopefully you have an idea for what is replication slippage. If you do not have any idea, I'll talk it about later on, because I don't have a picture of replication slippage. There's a beautiful picture of replication slippage in SNUSTAT. Very self-explanatory diagram of replication slippage. Anyway, by the mechanism of replication slippage, it so happens, see, the number of repeat is 6 to 59. Here, the number of repeats has increased to more than 200. The moment the number of repeats has increased to more than 200, you can see the length of the promoter region, and now you can see the length of the promoter region. With such a sudden increase in the CGG base pairs, what happens? Overall methylation takes place. So the CGI islands are no more protected here, and the whole of the CGI island and the promoter element gets methylated, as a result of which there is no primary transcript formation. This results in a disease known as Fragile X syndrome, where it seems that one of the X, so you can see the X chromosome. See, it seems as if this portion of the X is very much fragile. So that's the phenotypic expression. So this is one example how DNA methylation can be associated with diseases. This is another example. As you can see, in case of autoimmunity, we know that there are some autoimmune autoimmunity as a whole, most of them are congenital. That is, most of them to some extent are inherited. So the damage lies deep within. Now let us look at these two cells. Here the IFN regulated genes are in a compact form. That is, they are not automatically activated. But if you look into the case of the individual suffering from systemic lupus erythematosus, which is an autoimmune disorder, you can see the configuration of the chromatin. The chromatin is in the open form. As you can see that the histone molecules are acetylated. Now, what does this picture tell? This picture tells that since these chromosomes are in an open form, so the IFN gene somewhere sitting here is now always constitutively expressed, causing excessive inflammation. So this is basically most of the autoimmune diseases causes high amount of inflammation. So whenever there is a T cell stimulation, in normal cell we do not get the initial production of interferon gamma. Interferon gamma is produced only when the T helper 0 cell decides to go for a helper 1 phenotype. Okay, but here the moment there is a T cell stimulation, automatically, as you can see, the IFN gene gamma now no more regulated by the epigenetic modifications that were there, and thus they constitutively go on producing the IFN gamma IFN genes are constitutively expressed. And these genes again further control downstream a lot of another members of the cytokine families, which also includes variety of IFN subtypes, and thus a host of inflammatory molecules are released one after another. So this is an example of DNA methylation in autoimmune disease. And this is a case of another disease, which is PWS AGS syndrome, the Prader-Willi-Engelmann syndrome. Now this picture is from Strackman and Reed, not from the recent edition, but from the older edition. So I have showed you how the genomic imprinting to take place in the morning. Today it is a repetition of the same. Here you see this is a sperm and this is an egg. Now let's look at the two genes, A and B. Let us assume B is imprinted in sperm and A is imprinted in egg. Mind it, imprinting means either in most cases they are methylated, that is, they are silenced. So be kept in mind that these genes are not expressed, they are silenced by the phenomena of methylation. So let us assume. Now, so somatic cells, spermatogenesis. During spermatogenesis, erase old imprint. So the imprints are erased. So these are the gametes, the male. During the spermatogenesis process, see, in a male, the male has received one chromosome from the male father and one from the mother. The mother has A gene imprinted and the father has the B gene imprinted. So the somatic cell of that individual has two chromosomes, where in one chromosome B is imprinted and in one chromosome A is imprinted. This is the case of germ cells also. Now during spermatogenesis, sperms are produced. When sperms are produced, as you see, erase old imprint. Since both are to be male chromosomes, so if you erase the imprints from both A and B, and now establish fresh imprints on B only, because these are sperms. Sperms should have B imprints. See the egg. This is the daughter. Daughter has received the AB from the mother and the father. Now, this is going to give, this cell is going to give rise to an egg. So erase the old imprints, and both the egg cells just imprint the A. So you can see that this is how the imprints are erased during the process of spermatogenesis and oogenesis. Now let's look into the disease and the genetic mechanism that happens for the disease. Now what I was talking about, the Prader-Willi and the Engelmann syndrome. So let's concentrate. Let's look into these three genes, SNRPN, Nectin, UB3A. Now let us see that the darker colors are the imprinted genes. So this is imprinted this is imprinted. See, this is a female this chromosome has been obtained from the mother. Okay, let me see, this is chromosome 15 and the position is Q11. So this is from the female and this chromosome has been obtained from the father. In the female, the chromosome has these 3 genes. I mean, sorry, not female. The mother homologue has these 3 genes and the father homologue also has the 3 genes. Now, in case of the mother homologue, SNRPN and NECDIN are imprinted. So they do not give rise to any product. Clear? In case of the father homologue, the UB3A is imprinted. So there is no product. When we get a female, so this is a female and this is a male. As you can see, in female, the imprints are established. So in case of female, only the UBE3A is active. The other 2 are inactive. In case of male, only the UB3A is inactive, the other 2 are active. Right. So this is the condition for a normal male or a normal female. Now let us see that if there is a deletion in the sperm. So a sperm, so this is the father homologue. So a deletion in this region. So if a deletion occurs in this region, then the mother homologue will have these 3 genes, as you can see. Already SNRPN and NECDIN is imprinted, so no product. Only UB3A is giving a product, right? But in case of male, as you can see, these 2 are supposed to give a product. These 2 genes were supposed to give a product. But due to a deletion in the normal male, we are seeing that in the mother homologue, these 2 are imprinted. So we are getting a product of this gene. And just if we look into the opposite side, this is an egg with a deletion in this region. So this region of the egg is deleted. Now look at a normal female. So if this region is deleted. So for a normal female, this UB3A, both the UB3A are imprinted. So I will be getting this product. But when the female, this region is deleted, as you can see, the UB3A is imprinted and this region is deleted. So we are not getting any UB3A expression. So depending on whether the deletion is occurring in the male chromosome, so whether the deletion is occurring in the male chromosome or whether the deletion is occurring in the female chromosome, based on which side the deletion is occurring, we get a differential expression pattern of the proteins related. As a result, since in case of males, we get the expression of only UB3A, and in case of females, we get the expression of SNRP and NECDIN. So the 2 phenotypic expression of the disease are different. So in case of males, the disease is known as Prader-Willi syndrome, and in case of females, it is known as Engelmann syndrome. This is the example of the Prader-Willi-Engelmann syndrome and the example of differential deletion occurring in a male and a female chromosome and a male and a female genotype, as a result of which when deletion occurs, we get 2 different type of expressions. Now we are moving on to the next slide. So now I come to the last slide. This is a very famous case of epigenetic regulation with Drosophila as the example. Now, in case of Drosophila, it was seen that there are 2 competing groups of protein complexes, the trithorax group and the polycomb group, which are key regulators of epigenetic changes that are programmed during development. The name trithorax and polycomb refers to altered body parts associated with mutations in the genes that encode for TRXG, amino acid trithorax product, and the trithorax and the polycomb proteins respectively. Now, if we look at the molecular level, you can see that this is the polycomb response element. Okay, so this model actually considers the action of two different types of polycomb genes. Complexes which are named as polycomb recessive complex PRC1 and PRC2. So there is PRC2 and this is PRC1. So you can see there is a polycomb response element. There are two different polycomb complexes, PRC2 and PRC1. Now what happens? In the first step, as you can see, the PRC2 binds to a region known as the polycomb response element. The moment PRC2 binds to the polycomb response element, now let us assume that this is the target gene that is to be silenced by the polycomb, I mean to say this group of polycomb repressive complex 1 and 2. So this is the target gene. Now the moment the polycomb 2 binds to the response element, there is a first a polycomb response element binding protein binds, and then PRC2 binds. Okay. Now the PRC2 then catalyzes the attachment of methyl. You can see it results in the it actually activates methylases to add methyl groups to lysine 27 of histone 3. So you can see there are trimethylations. You can see there are trimethylations occurring in the histone 3 of the target gene area. So as a result of this trimethylation, so this is the target gene. As a result of this trimethylation, next what happens? PRC1 is further recruited. If you remember, I said that there are some methyl cytosine binding proteins. I just gave you the example that there are some methyl cytosine binding repressive proteins which further represses transcription, if you remember. So now once these areas are trimethylated, then what happens? The PRC1 binds here, and this further results in compaction of the chromatin. So three categories might occur. The chromatin can be compacted. Further methylation and ubiquitination can occur, or direct interaction with the, so this is one way, or direct interaction of the transcription factor with the PRC1 prevents the transcription factor from binding with the promoter elements. So this is what happens in case of Drosophila, and this is how this polycomb complex actually represses the target gene. And with this, I come to the end of DNA methylation topic. And for further studies, present knowledge you can get from reading papers. So after this basic concept, now you need to read some papers about how methylation is being used in cancer studies, how genome-wide methylation is being used for disease identification. So that you need to study much further to get more in-depth knowledge on the subject.